All right.
So the first few slides I'm going to show are sort of leftover things from that I either neglected to show or wanted to show and didn't show from last week, and then we'll go into new stuff. So this is a mammal. This is a primate, a cynomolgus monkey, and it's example of a exquisitely diurnal mammal with vision that's specializes in visual acuity. So just a couple things indicative of a primate. We looked at the ciliary muscle from a primate before, but it has this rather big ciliary muscle, and we talked about accommodation, how this posterior aspect of the ciliary muscle drags this soft tissue forward and closes this area, making the aperture, which supports the lens smaller, which means that the zonular ligaments become less tense, which provides an opportunity for the lens capsule to cause the lens to be rounder for near vision accommodation. But I wanted also to show that this is Schlemm's canal. Most of the animals that we looked at that are of interest to veterinary species, rather than have a Schlemm's canal, they have an angular aqueous plexus. Schlemm's canal is characterized by being circumferential around the globe. So no matter where you cut it, you'll always get a cross section of the Schlemm's canal. It's a vascular structure that handles the egress of aqueous fluid from the eye. So from here, it goes through a series of vascular channels that eventually ends up in the general circulation. But under normal circumstances, all of that drainage, all of those drainage angles are filled with aqueous and not blood. In the angular aqueous plexus, rather than being circumferential, is intermittent. So you won't necessarily always section it in every section, and sometimes you'll get more than one aspect. Well, what I wanted to show this for is to show in the back of the eye. The fovea which is here, and let's just go to higher magnification. So the fovea, as you probably know, is the portion of the retina that's most useful for a high degree of visual acuity. In people, when you're looking directly at something, you're focusing your image on the fovea. And the way to know if you're in a fovea is to look for ganglion cells, and the fovea will be rich in ganglion cells, as you can see here. And then the other thing to let you know if you're in or around a fovea is an absence of a nerve fiber layer. The nerve fiber layer emanates from the fovea. It doesn't pass over the fovea. And then in primates, it's an area of concentrated cones, because cones are the cells that are responsible for visual acuity. And I can't really show you that these are cones on histology. And then if we go from here and go towards the optic nerve, you can see that the nerve fiber layer, which is here, is going to get thicker and thicker. And eventually, when we reach the optic nerve, the nerve fiber layer will turn in, and those are the axons of the ganglion cells, and they will proceed within the optic nerve up to the brain. So lots of ganglion cells. If we proceed from here to go to the more peripheral retina, you'll see the number of ganglion cells will decrease. It starts to look like other animals. The other thing about using humans as an example, if you're looking directly at something or centered on something, you'll be using your fovea. We're not actually that aware of our peripheral vision. People can lose their peripheral vision and they will not be aware of it. That's basically what happens in glaucoma. But the actual visual acuity of peripheral vision is not bad. If you think of something like a point guard in basketball, or someone who needs to know what's happening around them, it doesn't take, or just think of looking and maybe something like an insect, or there'll be some motion in the periphery, and you'll become aware of it, but you won't necessarily be conscious of it. What you'll do then is turn your head in order to identify what it is.
You mentioned yellow color of the macula, right?
Yep, macula lutea, yeah.
And what pigment is it, and where exactly is
It's
it?
lutein, and it's in the inner retina, and you can't see it histologically.
Oh, really?
You can see it.
Huh.
If it's there, you can see it grossly sometimes.
Okay.
We have some pictures of orangutans and
Mhm.
gorillas in which the macula lutea is
Lutein, what is it?
pretty clear. Pardon me?
Lutein, what is it? What is lutein?
Oh, what is
Lutein? Oh.
Yeah. And why is it there?
Oh, so that's a good question. If you see yellow in the eye, either the lens or the retina, good bet that it's there for protection against UV light. And what is lutein?
Isn't it a vitamin of some sort? I mean, it's
Xanthine, lutein. I don't know.
The carotenoid group, apparently.
Right.
Isn't that included in the supplement with the ARID study?
It is.
Yeah. Okay.
Oh,
It's antioxidant.
So if you buy vitamins that are
Yeah.
supposedly good for your eyes, they usually have lutein.
it says it may help prevent or slow macular degeneration. I think it's a very quick Google search.
They inner retina, right?
It's in the retina.
Is it water soluble? That's why, well, no, because then it would be in the, never mind. Okay, I'm giving up. Why can't we see it microscopically?
No.
It should
And
be
then-
the cells which produce this lutein, right?
This what?
Should be the cells, probably cones itself produce this lutein.
No, I don't think so. I think it's in the inner retina. And it's UV protection.
They must accumulate it from the blood somehow. As opposed to like making it. All right.
Julie Mares,
Okay.
who's retired now, spent her career studying lutein,
Okay.
but she's not here. Okay, I showed you a picture of a diurnal raptor, and I mentioned that they have not one but two foveas. And so I was hoping that I could get a slide from Switzerland, from the researchers that produced that beautiful picture. This is one that this is a red-tailed hawk. It's our best attempt to get a section of the raptor retina. So most birds, as I've said before, most birds have a fovea, which is you can see here. I shouldn't even say most birds. Many birds have a fovea. I don't know if it's more than 50%. And then some birds, relatively few birds, but all of the diurnal raptors, the eagles, hawks, falcons, and there's another one that I'm forgetting, have not one but two foveas in each eye. So this is our attempt to sample both foveas in a red-tailed hawk.
Are they visible grossly?
That's a great question. I think because of the nature of the vitreous, that it's hard to recognize that bump. Otherwise, we'd probably try to get them more often.
Okay.
I remember Billy and I spent maybe an hour in an eagle eye looking for the fovea and trying to pull the vitreous out, and
Yeah.
never were able to find it,
Hmm.
which is embarrassing to me. But same thing. The ganglion cells are most numerous here. The neurofiber layer is absent. The inner nuclear layer in birds is thicker and more complicated than you'd find in a mammal. And I don't, I'm not a neural processing person, but I'm told that that the neural processing occurs to a greater extent in the bird eye than in the mammalian eye. So
Can I
that's
ask a quick question?
sure.
So is this not exactly in the center of the fovea because there's still an INL?
Internuclear layer.
Yeah,
No, there's no INL. Here's some INL.
well, that's a ganglion cell layer.
No, here's the ganglion cell.
Yeah.
Here's the the internuclear layer. So I was thinking the nerve fiber layer.
Okay,
Uh,
yeah, the internuclear layer. So does this mean that we're not exactly in the center of it because we still have quite a thick internuclear layer there?
don't know.
Okay, all right.
There's almost a certain chance that we're not directly in the center of the fovea, just because who would know? How
Right now, your hand is the ENL in the INL.
Yeah.
Nuclear layer.
Yeah,
That's for bipolar cells, right?
exactly bipolar cells,
horizontal.
Mueller cells, horizontal cells, amacrine cells.
And what did you say that about central retina and inner nuclear layer?
So in the birds that have two, there's a central fovea. If the bird is looking directly at something, then it has stereopsis because the central fovea will be used in both the right and left eye. And there's also a temporal fovea, which is specific to the eye. And you'll see birds sometimes kind of eyeballing something. And when they're doing that, they're using the temporal fovea. And I don't know which, I think this is the central, but I'm not sure. So we're going to go,
How to detection of moving objects or
I'm sorry?
to detection of moving objects, because that's how they hunt, right? That's the purpose of the eyes. They somehow
So,
able to detect
They have incredible visual acuity. I'm not saying they can't detect motion, but I don't think their eyes are specifically adapted to detect motion. It's just incredible visual acuity. So there should be,
there's a nice figure in there.
yeah,
about
let's
the-
put that figure up with the multifocal.
Oh, this guy.
Yeah, this shows the temporal fovea is the same in both eyes, so it has stereopsis. And the central fovea is specific to each eye, and then go to the one before that. So this is the the section I was hoping to get. This is Petra and Simone did this research and got these beautiful sections, including both fovea. But this is work that was done here. This is a multifocal ERG. Do people know what that is? You know what ERG is, an electroretinogram. Electroretinogram converts the electrical activity of the retina to a flash of light into a graph that shows you the amplitude of the electrical activity. And in a human with a multifocal ERG, the amplitude of the response in the fovea creates this mountain. This is a horse.
That's kind of laughable. The horse one.
Yeah,
It's like, what do they
horse
see?
one. Well,
Nothing?
Move so easily, it's in shadow.
we know histologically.
I guess.
Horse has such a huge eye, but
I guess it's
it is kind of laughable. But this is the red tail. Jim Verhoeven as an electrophysiologist at the medical school, and he was able to get one of these in a red-tailed hawk, which is very suggestive. But who knows what, this is just the amplitude of the electrical activity. It doesn't measure function. Okay, so let's go back to the slide. So now we're moving along the eye. Notice that the nerve fiber layer gets thicker and thicker. This area right here, ganglion cells get fewer and fewer. And then we're going to come along here and the nerve fiber layer starts to get thinner. Ganglion cells start to get more. And then we are near, but not in the second fovea here. And this has an even more robust, internuclear layer.
It's neat how you can actually see some stratification in the internuclear layer.
Yes.
Well, and in the outer nuclear layer. Whereas like in dogs and cats, there's like nothing.
Yes.
Like I guess we can sometimes see the cones,
Yes, I wish I knew what that meant,
but yeah, I'm sure
but I don't.
there's a ton of research as to what types of cells are making up the different strata.
It is very interesting, but I don't know. Petra and Simone did OCG on these. I don't think they made any conclusion about the stratification of the
Mhm.
nuclear layers, but
Cool.
they've written a paper on it, so it might be there. Okay, now we're going to cover something I also meant to cover last time. And we're going to start out with and this is in a bird, and we're going to start out in a hummingbird. And we're going to talk about, have people watched bird eyes and noticed the movement of the nictitans of the third eyelid? You know, is there anybody here that doesn't know what I'm talking about? So birds, like many mammals, have a third eyelid, but it's arranged a little bit differently. And the action of the third eyelid is extremely fast in a bird. So this is the head of a hummingbird. Here's the eye. This is the lower lid, and this is the upper lid. The lower lid is always the more dominant lid in a bird, and it's also unlike in mammals, the upper lid is the one that opens and closes. In birds and reptiles, the lower lid is most often the one that opens and closes. Here's the nictitans. Here's the third eyelid, and then if we go to the back of the eye. So the action of the third eyelid in birds is accomplished by skeletal muscle, and it's accomplished because of this tendon here. This is the pyramidalis tendon, and this muscle that's connected to the sclera is the quadratus muscle. And I'll show you a picture that shows that a little bit better. And then you notice that the tendon is in a tenosynovium. It's in an area where it's free to move back and forth. So we can pick up that tendon again over here. Oh, I think we need to go to this side. And we'll pick up that tendon again here. Did I show this one already? Here it is again. So that tendon will wrap around the eye. It'll grab the third eyelid, and when it contracts, that third eyelid will race at lightning speed over the cornea. And we'll come back to what the effect of that is. So why don't you put the picture? So this happens to be an eagle, and I've false colored the tissues involved. Here's that quadratus muscle again, and I've opened up here's the tenosynovium that I showed in the hummingbird. I've opened it up, and then here's the pyramidalis muscle. So the action of the pyramidalis tendon depends on these two skeletal muscles, both of which are attached to the sclera, and they're both innervated by the abducens nerve. They're the equivalent of the retractor bulbi that we see in some mammals. And so the tendon I false colored green, and we pick it up on the other side here, comes up underneath the eye, and spreads out over the nictitans, which is here. And so then when that contracts, the nictitans goes flying over the cornea almost so fast that you can't recognize that you've seen it. Okay, next picture. So here's a histo picture of the nictitans with an Alcian blue PAS stain, and stains both. This is the third eyelid again in a different section in a different bird. And what I want to point out here is that the epithelial cells on the on the bulbar side of the third eyelid have these arborizing cellular processes, and those arborizing processes are populated by mucins. This is an Alcian blue, so it's staining the mucins with Alcian blue, and then by electron microscopy, this is what they look like. Dr. Schwab calls these the feathered epithelium, and I haven't seen anybody else describe them, so I just call them a feathered epithelium. What they do is they imbibe themselves with the mucinous aspect of the tear film, and then when they spread over the cornea, I think of it like a roller brush for a painter. It actually deposits the mucinous elements of the tear film onto the cornea. Also cleans the cornea at the same time. Any questions about that, the third eyelid? When you have the opportunity to look at a bird, look carefully for the third eyelid blink. Okay, so now, sticking with birds, I'm going to show you, this is a great horned owl. So it's a the default function of a bird eye is to use to be functioning in the daylight with plenty of light, and therefore be a specialist in visual acuity. And then, of course, there are some exceptions. There are nocturnal birds, and the great horned owl is a great example of that. There are diurnal owls, so I'm not going to say this is totally representative of the owls, but it's a great example of a large owl eye. So the owls have what's called a tubular eye. They have an elongated, almost rectangular eye, and the reason why they have that is they have this gigantic scleral ossicle that goes from almost the back of the eye to the front of the eye. And other than that, they have features typical of birds. Here's the pectinoculi. Here's that gigantic scleral ossicle. Let's go to the other side. The first slide was better. They have a very robust annular pad, which is kind of shattered in this particular case. You can see the ciliary processes attached directly to the equator of the lens. They have they have a robust Crampton's muscle. So remember that birds have skeletal muscle in their uvea, and there's an anterior and a posterior. This is the anterior muscle. It's the Crampton's muscle, and Crampton's muscle is responsible for the changing the curvature of the cornea for accommodation. So it'll increase the curvature of the axial cornea. And then the great horned owl, characteristic of birds, of the nocturnal birds, is devoid or only has at the very most a very rudimentary posterior ciliary muscle or Brooks muscle. So accommodation in the nocturnal birds, that includes the owls, the things, the nightjars, things like whippoorwills and nighthawks, all are similar in that they lack a Brooks muscle. I don't know. I'm not completely sure why that is, but I think it's because this massive bone prevents the action of Brooks muscle from closing the aperture, pulling this tissue forward, closing the aperture of the cornea. Oh, and then let's look at the retina. This section doesn't show it, but the great horned owl is one of the very few nocturnal animals that has a fovea. And there's not too much, we'll see some reptiles Remember that birds are reptiles, in which the outer the outer segments of the photoreceptors are very short. But in the owl, I'm not going to say that they're a different category of photoreceptor than the mammalian rod, but they're rod-like in their morphology, hence their function in dim light.
They look like they still have pretty high retinal ganglion cell density.
Yes, they do. When they have a fovea, they have remarkable visual acuity, especially for dim light vision, and they have they're one of the few nocturnal animals that has a fovea. Okay, now we're going to talk about snake eyes. So now we're going to go through a potpourri of reptilian eyes. Remember I said that birds are related to theropod dinosaurs, and we don't have theropod dinosaurs anymore to look at. Presumably they have eyes an awful lot like birds. More closely related to birds are crocodilians and turtles. Turtles are somewhat questionable, and we'll look at a crocodilian eye, but not now. Then much more distantly related to birds are your various forms of lizards. And snakes are, although they don't look like lizards, they're genetically similar to iguanas, monitor lizards, geckos, and chameleons. I'm calling all of those lizards. So this is a snake. This is a ball python. And it's a decalcified head of the snake, so it's more than just the eye. So here's the back of the eye. Here's the optic nerve. The retina is artifactually detached. Slide is coming. It's pretty dirty. I said in the beginning that only mammals will have blood vessels actually in the eye. And I think I showed you a fish that had a membrana vasculosa. And here's the membrana vasculosa in this snake's. Here's where the blood vessels are. So they're the blood vessels are contained within the vitreous. They're not within the retina. Only mammals have blood vessels actually in the retina. But if you did fundoscopy on the snake, you would think that they have retinal blood vessels. They would look similar. Snakes do not have cartilage in their sclera. They don't have bone. So somewhat like mammals to that degree. They have skeletal muscle. All the reptiles, and only reptiles, have skeletal muscles in their eye. They don't have big muscles for accommodation like I showed you in the bird.
Crocodile, right?
No, this is a snake. This is a ball python. But the snake,
Sorry, do they have Pardek or Meshwork? I cannot see from this slide.
it has snake morphologists will name this structure. And I think we can see it better on the other side. Not really. Snake morphologists will name the structure right here, Schlemm's canal. And I don't know if that means it's exactly analogous to Schlemm's canal.
It looks quite similar.
Yeah. They don't have plica like a mammal would have, but they have this roll in the ciliary body. They have skeletal muscle. But the thing that I want to point out is that they have a double covering of their eye. So here's the cornea. So this would be the anterior chamber. Here's the cornea. And then they have this second covering of the eye called the spectacle. the spectacle is continuous with the integument, with the skin. And then the cornea has this fluid-filled space between the spectacle and the cornea, which is the subspectacular space, and that is filled with fluid. It's lacrimal fluid, and if we follow this back, you can see that this fornix goes way back in the eye, and this would be the equivalent of the fornix of the conjunctiva. And then if we go a little bit further, here's the lacrimal gland. We haven't sampled the ducts of the lacrimal gland.
What's this thing?
That's a muscle. So let's go back to the cornea. Notice that the cornea is lacking in epithelium. If you have really sharp eyes, you might be able to see that there's a very attenuated epithelium. And there's also a very attenuated epithelium on the back of the spectacle. So can you suggest why that would be? Why is the snake cornea lacking in epithelium?
Does it need the barrier function there?
Right, that doesn't need a barrier function because it's bathed in fluid continuously. The epithelium is there. If this spectacle were to be damaged, or if there was some inflammation, that epithelium would regrow rapidly. But if we follow it back, also the epithelium on the back of the spectacle would proliferate and regrow rapidly. I think, as you probably noticed when I did this before, the epithelium becomes more and more apparent as we go back into the fornix, and you can also see it has a secretory aspect as you would expect a conjunctival epithelium to have. So we have the snake head because it was part of an experiment. Dr. Chris Murphy and I wanted to study the morphology of the healing of the spectacle. So we got a series of ball pythons, and we surgically excised a segment of spectacle. And then we sacrificed snakes at various periods of time to study the process of the healing of the spectacle. And this is a zero-time control. So here's the surgical site. There was no time for any cellular reaction to occur. So everything is as you'd expect it to be in the native eye, except that there's this surgically induced defect. Now, the spectacle in a snake has blood vessels. You can see blood vessels here. So nutrients can go. And then, so the pathophysiology of the spectacle becomes important because there are conditions in the snake around the time of shedding of its skin. The medical term for that is ecdysis. There can be complications in snakes related to their eyes associated with an incomplete or poor completion of the shedding process called dysecdysis. And it's very common to say that the spectacle sloughs during ecdysis. It's also common to say that the skin sloughs, and neither one of those is true. If the spectacle sloughed, then the cornea would be very badly damaged. I can't remember how many snakes we did in this experiment, but at least one of them had to be put down because the corneal pathology was too severe, and almost all of them had pathology. All of them had a proliferation of the corneal epithelium and also the posterior spectacle epithelium. But the only part that sloughs is this completely acellular, completely keratinous layer known as it's kind of stupid name, which I should have memorized before I got here, because I always forget it. It's the I like to call it the cuticle. I think that's a much better name. So I'm just going to go with cuticle here. But this is the only area that sloughs. And then in the skin, the only area that sloughs in the skin is this, the equivalent, the same totally keratinous, totally acellular area. But in the process, most of the life of the snake will have a clear spectacle, no blood in the blood vessels of the spectacle, a clear cornea, and the snake will be comfortable. As ecdysis approaches in the snake, the blood vessels will open up, and blood products will leach out into the spectacular stroma, making the spectacle cloudy. And during that process, I thought maybe I would catch it if I a new outer generation. A new outer generation. I told you a stupid name. A new outer generation will form on the surface here, and ecdysis won't occur until that new outer generation is complete, and then ecdysis will occur. A few days before ecdysis occurs, the blood will leave the blood vessels. And the other thing that happens when the spectacle is cloudy is the snake becomes irritable. And a few days before ecdysis, the blood will leave the spectacle will become clear again, and the snake will become calm, and then ecdysis will occur. If you have dysecdysis, there's some sort of anatomical problem here, usually associated with hyperkeratosis, but this separation will not occur completely. And then there are recommendations for coaxing that attached abnormal epithelium off, and that's not my department. So this is a ball python. This next one is a rattlesnake, and Megan received this snake, and she decalcified the head. And then let's put it on here. She decalcified the head.
We do go to 4.
And she did what us pathologists call a bread loafing. And the head was cut as you would a slice of making slices of bread. You cut through the head as if it was a loaf of bread. So we're going to look at the section that has the eyes.
The baby.
Hey, what?
It was a tiny little snake.
Don't get bitten by it. And now we got to find that section we're interested in. That's the trachea. Okay. So I think this one is better. Now we're going to look at the spectacle again. A little bit more of a recognizable epithelium on the cornea here. And the stroma is much thinner. I don't know what the significance of any of this is, but the stroma is much thinner than in that python. And then the outer generation is ready to slough. And now you can see there's a inner generation at least partially intact, allowing for ecdysis to occur. That's the main reason I wanted to show this.
Space between that outer generation and the spectacle, is that artifact, or are those actually touching? each other?
No, it's almost impossible to get a section of a snake eye that doesn't have a space between the outer generation and the spectacle, suggesting it's tempting to think, well, it's undergoing ecdysis, but that doesn't seem to be true. It just seems to be an area that artifactually separates in the processing. This is actually the venom duct, and it's got a venom, what's the word for inflammation of a duct? Ductitis. So pythons and vipers, this rattlesnake is a viper, have a mechanism for detecting infrared light. It's called a pit. And here is this is a section through the pit of this rattlesnake. The detection of infrared light, heat in other words, is registered in the brain in the same way that visual light is. It's registered in the visual cortex of the brain. It's directional because it goes through a pit. So a snake can detect the direction of where the heat is coming from, but it's not. I wouldn't think of it as vision. There's very little acuity or spatial recognition, but it's quite simple in its construction, and it's well illustrated here. There's a vascularized membrane, almost like a tympanic membrane. It goes over the surface. Then there's a very thin cavity beneath that. And then on the head side, there are these little bumps. And it's within these little bumps that there are sensory cells that register the infrared detection. And don't ask me anything more about that than that. It's just a byproduct of sectioning that snake.
You mean the amazing talent, of Dr. Klimans?
You betcha. So spectacle. Now we're going to do a potpourri of reptiles. And the next thing we're going to talk about is geckos. There are lots and lots of different kinds of geckos around the world. I don't think we have any in North America, but they populate the world. Most geckos are nocturnal predators, which is unusual for reptiles. Reptiles by nature tend to be diurnal and have eyes that are adapted for visual acuity. And then most geckos, like snakes, have a two-layer surface of their eye. So they have something like a spectacle. It's I think more correctly in the geckos referred to as a brill, and I'll show you the morphology of it before, but people also call it a spectacle because basically it's the same as what a snake has. Then a second group of geckos are what are called eublepharic geckos, and those are geckos that have true eyelids and do not have a brill. The most popular pet gecko is the leopard gecko, and that happens to be a eublepharic gecko. It has true eyelids and does not have a brill. I'm not going to show you any eublepharic geckos, but I'm going to show you this. I've forgotten what it is. I have it labeled on the back here. This is that's a ball python. This is a Madagascar leaf-tailed gecko. And I've looked it up, and this is a nocturnal gecko, as most geckos are. And we're going to talk a little bit about nocturnality in the gecko as well. If you have a gecko and you ask yourself the question, is this a nocturnal gecko or a diurnal gecko? You can tell by looking at the eye, because all of the nocturnal geckos will have a slit pupil. And if I don't have a picture, but if you're familiar with gecko eyes, the geckos will have a very pronounced slit pupil it closes basically all the way, and then there are little pinholes up and down the aperture. And the optics of that is way over my head in terms of the mathematics of it, but there are apparently functions derived from the multiple pinholes of the gecko pupil. But I'm not going to talk about that. You should. Again, this is the decalcified head. Here's the eye. And we'll start right out with the brill or spectacle, if you will. And again, you can see there's a two-layered spectacle here. Now, this is not a snake, so there is an annular pad in the lens. There's a bone in the sclera. There's cartilage in the sclera, just as there should be in a reptile. I guess we haven't sectioned it appropriately, but it should have a vascular structure, the conus papillaris, which comes out of the optic nerve and extends into the eye. So it has all the features that we expect in a reptile, unlike the snake. Here's the cornea. And you can see, now we can recognize attenuated epithelium, but there is definitely an epithelium on the cornea, unlike the snake. The gecko cornea, oh, yeah, no, I'm back off. So here's the subspectacular space, or maybe we could call it the subbrillar space. I'm not sure. And again, it extends back into what would be analogous to the fornix of the conjunctiva. The epithelial nature of it doesn't quite approach what we see in the snake. I'm not sure why that is. And then over the top, we have the brill or the spectacle. And again, there's this completely acellular keratinous layer. I'm not sure if that's called an outer generation in a gecko, but this is what's sloughs during ecdysis. Geckos can have similar dysecdysis problems as snakes, and that acellular keratinous layer is also sloughs from the surface of the integument. The gecko spectacle does not have blood vessels. So it's a little bit different in the cycle of ecdysis. Okay. So I mentioned once about how to differentiate, looking at just the retina, how to differentiate an animal that's it has a high degree of visual acuity and sees in the bright daylight, and animals that have a high degree of visual sensitivity and see in the dark. Basically, those that see in the dark have a thick outer nuclear layer because they have mostly rods in their retina that take up less space, and then a thin ganglion cell layer because they a large number of rods will hone in on one ganglion cell. I think I mentioned when I said that rule works well in mammals, but it doesn't work very well in birds and reptiles. Remember that birds are reptiles. It works pretty, you might say, okay, well, you just showed me an owl. It works kind of well in the owl, and nothing about morphology is universal. But this is a nocturnally hunting gecko, and you can see here's the outer nuclear layer, and it's a very thin outer nuclear layer. To look at these outer segments, someone who's used to looking at mammalian retinas would say, oh, those are all cones. So they get to the crux of it with nocturnal reptiles. And I don't know that this is true for all nocturnal reptiles, but at least some nocturnal reptiles, the nocturnality, the ability to see in dim light, in other words, sensitivity, is based on the fact that they have repurposed their cones. Remember, these are derived from, derived by evolution from animals that were basking in the bright sunlight and developed visual acuity in the bright sunlight. So they developed cones like any other animals, but they repurposed those cones with similar morphology to be sensitive to dim light, which I think is fascinating. One of the fascinating results of that is that the gecko, I don't know about other nocturnal reptiles, but the nocturnal gecko maintains color vision in dim light, because these are the same cells just repurposed to have vision, to be more sensitive to in dim light. And again, geckos, in evolution, geckos go way back earlier than any of the other lizards. And that is to say, the fossil morphology of the gecko goes way back in time. So they have lots of time to develop eublepharik and geckos with brills, nocturnal and diurnal geckos. So that's the gecko. Any questions? One time I attended a meeting of eye pathologists, physician eye pathologists, and we were all gathered together in an informal lunch, and I was with a group of them, and they asked me, well, what's my favorite eye? And I said, well, every eye is unique, and I don't have any particular favors. So they would have nothing of that. They insisted that I name an eye that was my favorite eye. And the eye that I came up with, and I think I'd stick with it, is the chameleon eye. Is anybody familiar with how the chameleon eye works? If you're not familiar with anything else, you might know that the chameleon has individual function of the motion of both the left and right eye. So you have to look at me to see the motion here, but their eyes will rotate back and forth. The left eye in a different plane than the right eye. I gave a talk on chameleon eyes to the European veterinary ophthalmologists, and the neurophysiologists in the meeting had a hard time dealing with this independent motion of the eyes. And the other thing about chameleons is they feed by catching insects with their tongue. And I've never tried to catch an insect with my tongue, but I think that it probably involves a high degree of depth perception, a very sensitive depth perception. So the other thing that's most fascinating to me is the chameleons are the only vertebrate eye that I know of that have a negatively powered lens. All other animals have a positively powered lens. So the curved cornea and the lens work together to form an image, to form an in-focus image on the retina. So if you have a negatively powered lens, what it does is it makes the optical center of the lens of the eye move forward. And what that means is that all of the refractive power of the eye has to be in the cornea. So they're able to create a remarkable curvature of their cornea, and I'll show you a little bit how that is. They have a remarkable annular pad. They have cartilage. I think you can see just a little bit of the conus papillaris. So all of the features of that you'd expect to find in a reptilian eye. Here's the scleral ossicle. This is the lower lid, and this is the upper lid. Remember, the lower lid is the more dominant lid in reptiles. If you rely entirely on Google, you can be led to think that the conjunctiva and the opening of the eyes. What's the opening of the conjunctiva called? The
Fornix?
no, not the fornix, the
Palpebral fissure?
palpebral fissure. You'd be led to believe that the palpebral fissure is attached to the limbus, but that's not what I see. I see a fairly deep limbus, and it looks to me as if the eyelid moves with the eye because of a complicated arrangement of muscles here. But I'm not willing to say to go overboard on that. But here's the palpebral fissure. And let's look a little bit more magnified. Notice that you can almost not see the. Oh, that's not going to work. So here again is the scleral ossicle. Here's the iris, and the chameleon has an extremely thin cornea. And I think that explains to some extent why it's capable of achieving a dramatic axial curvature. You can't tell on this histo slide. I wish there was a way, either in the gross eye removed from the head or on histology, to calculate what the corneal curvature is, but it's hopeless. But I think this very thin cornea will allow that cornea to achieve a remarkable axial curvature. And we look at this. Here's a muscle, a skeletal muscle, and that muscle is attached to a long collagenous structure, which we'll call a tendon. And that tendon goes up and attaches to the innermost peripheralmost cornea. So I think when that contracts circumferentially around the eye, it pulls the innermost cornea in, just like in a bird, and then causes the axial cornea to undergo a dramatic curvature. I showed you in birds how there's lens accommodation. At least looking at the musculature, I'm not sure how the chameleon would achieve lens accommodation, but maybe there's some structure that I don't understand, but it has a dramatic annular pad. I suspect that dramatic annular pad is somehow related to the formation of this indentation in the front of the lens, which gives it a negatively powered lens. And then I'm going to go to the I have another section of a chameleon, which is more centered on the back of the eye. You sure? Won't break it.
It's okay, still intact.
And if you look at the back of the eye, the chameleon retina, the peripheral retina is here. The peripheral retina on the other side is here. And it's really nothing more than a very short, very prominent fovea. So remember, I told you, if you're going to catch an insect with your tongue, it's probably very wise to have a high degree of depth perception.
Hmm.
So, this fovea is important, I think, for depth perception, but it's also only a small portion of the retina. So the chameleon has tunnel vision. It's able to see only a small part of the visual field. And the chameleon is both predator and prey. So for predation, it needs that depth perception. To prevent predation, it needs to be aware of what's going on around it. And I think that explains why it's able to move each eye is independently, constantly looking at different areas of the environment to be on the lookout for predation. And the gecko also has macular lutea. If you look at the gross section of a retina, there's a pretty bright red, a pretty bright yellow coloring to the back of the fovea area.
In this fovea, the outer nuclear layer is discontinuous, which seems counterintuitive.
This
Shouldn't there be photoreceptors in that pit?
I'm not
I
the
mean, I
one-
guess there are outer segments. There are outer segments.
I
there.
won't claim to be the one who can answer the question of whether there should be or shouldn't be something. I agree with you. It's discontinuous there.
Well, I mean, I guess the photosensitive part is there, just
Yeah.
not the nuclei. Then they must have really long inner and outer segments or something.
Yeah.
Oh, okay. Weird.
Any questions about the gecko? Try and catch an insect with your tongue.
Well, if we had a tongue like a chameleon, it'd be
Yeah.
a snap.
We have a nice section of a midline head section that shows the morphology of the tongue. Quite interesting. So this is a leaf turtle. I don't know what a leaf turtle is, but that's what this eye is from. And we're sampling it here as a generic turtle. Is that it? Oh, there's two sections in here. Hopefully the other one is better. Well, not much better. So turtles have cartilage. They have bone. You can see the scleral ossicle here. I don't know if they have a fovea or not. This section doesn't prove it, but they do not have a conus papillaris. You have to take my word for it. It's not shown here. They have a very rudimentary
Missing the cornea here artifactually, right?
right
Okay.
annular pad, very thin one. So I don't have too much to say about turtle morphology. They have a very robust musculature in their iris. I'll say just a word about that. You're right, no cornea on this one. There's a second section in here. Maybe we have cornea there. Yeah, we have some cornea here. They have oil droplets. They have double cones. They have all the other features that a reptile has. Paleontologically, where turtles fit in the phylogeny of reptiles is unless it's been clarified recently, it's quite controversial. Most phylogenies put the turtle along with the crocodiles as being part of the group that evolved from more primitive reptiles to form dinosaurs, and eventually birds, but that's quite controversial. Anyway, more primitive reptiles like the lizards that we showed have a nice prominent conus papillaris. Turtles don't, and I don't have much more to say about that. It's confusing. Let's look at this iris. Only thing I'm going to say about the iris here is that it's got a robust skeletal muscle stroma, and I suspect, because of other specimens that I'll show you later if there's time, I suspect that this is somehow related to an animal that would like to have vision both underwater and above water, and then it's related to the ability to dramatically change the curvature of the anterior lenticular surface. I don't know, I've never seen that, but I'll show you some birds, some diving birds that have similar morphology. This now is a sea turtle. This is just says sea turtle. And there's not too much I want to show in here. Well, let's do it this way. Again, there's no conus papillaris. There's a scleral ossicle. There's quite a prominent scleral cartilage. And then, I don't know why, but the sea turtles tend to have a very small, very round, very anteriorly positioned lens. And I've never pursued or tried to look for a manuscript that describes why that is. We have sections of several sea turtles, and it seems to be a phenomenon. We don't have that many crocodilian eyes.
Do you know if the sea turtles have any adaptations for deep diving? Sea turtles go that deep for the pressure?
So the pressure, when you talk about adaptations of the eye for animals that dive deep, everybody always jumps to the pressure, but it's a fluid-filled structure with fluid on the inside and fluid on the outside. Pressure shouldn't have much of an effect on the eye. I believe you can take a grape and put it in the ocean at any depth you want, and the pressure it'll remain a grape because it has fluid on the inside and fluid on the outside. When we get to cetaceans and pinnipeds, seals, I'll show you some other adaptations that people always jump on and say, well, that's because of their adaptation to pressure. But I don't think that's true. I think the pressure itself has a little effect on visual function. So this is, I've forgotten what, oh, this is a gharial, which is a crocodilian. Let's go to the subgroups. So the crocodilians, again, have a robust cartilage in their eye. They have no conus papillaris. They have no bone in their eye, so sort of like a mammal. They have no annular pad. So many of the features that we see in reptilian eyes are not here. And yet, pretty clearly, unlike the turtles, the phylogeny and paleontology of the crocodilians is not complicated. They are clearly derived in the lineage that became dinosaurs and eventually became birds. So I think that it's reasonable to suspect then, since more primitive reptiles have those structures, crocodilians don't have those structures, it's reasonable to presume then that those structures have been lost in the crocodilian eyes. And in support of that, crocodilians are exquisitely adapted for nocturnal vision. They have slit pupils. They have many of them have a tapetum, and so just like mammals, I think it's reasonable to assume that they have lost somewhere along the line. They have lost some of the sophisticated structural characteristics of animals that develop high degree of visual acuity. So yeah, I think I'm gonna try to show one more thing in this gharial. Nice reptilian photoreceptors. Notice a very scantily populated outer nuclear layer. So again, this is a nocturnal animal. The photoreceptor cells have repurposed themselves to be sensitive to dim light. Now, I think this will work. We're going to try polarizing this. Oh, it's been in the whole time. Oh. We're going to try moving it around and see if we can find an area. I thought this
Can you
showed.
look in the thing here and see? if it's- Right.
I remember this as having a guanine.
Something's not working. I
Oh.
feel like
Can you see it through the pieces?
it never gets really black. Shouldn't make it really black. It just turns really pink.
Isn't that something the computer does?
Well, I'm looking through the eyepieces though.
Oh.
What do you think, Kelsey?
I mean, yeah, I see pink.
Okay.
I'm seeing the square, the collagen though does polarize.
Okay, well, I'm gonna abandon that.
Yeah.
What is, oh, sorry about that.
Let's actually pull the upper polarizer out now.
Yeah. Okay, I was hoping to be able to show a retinal tapetum, and I thought this gharial had it, but I should have checked ahead of time. Okay, so that was a potpourri of reptilian eyes. Are there any reptilian questions? Yeah. Yeah. So yeah, a lot of animals more primitive than mammals will have a guanine tapetum. Guanine is a amino acid that can crystallize, and when it crystallizes, it gives you a silvery appearance. If you look at a tuna fish or a fish that has a silvery outside, that's because they have a guanine layer in their integument, and so they'll have a tapetum that's made up of guanine. And I thought I could show that it polarizes really nicely. I thought I must have in mind some other specimen. Okay, any other questions about reptilian vision? The thing to know that I think the take-home message is reptilian vision, and in terms of anatomy, reptilian and avian vision is related phylogenetically, although there is marked differences. Now we're going to talk a little bit about fish. And I'm going to show you this is a flounder. Nothing special about it being a flounder, it's just a good fish eye. So can anybody speculate about how vision underwater is different than vision, terrestrial vision in the air? The what?
So the light doesn't have to go through air and then water into the eye, so then the lens shape tends to be very spherical.
Right, I'm not sure I completely follow you, but in terrestrial vision, in order to form an image, in order to focus light on the retina, there are two structures that participate in that refraction. One is the curved cornea, and the curved cornea, remember, is a corneal air interface, and the other one then is the lens. So they will function together to form an image on the retina, and the degree to which a tissue is capable of undergoing refraction for focusing purposes is dependent upon the curvature, for one thing, and then the second thing is the optical density of the media, optical density of the air, optical density of the cornea. And in that situation, in the terrestrial vision, there's a considerable difference in the optical density between air and cornea. So that means that the cornea can participate in forming an image on the retina. Underwater, the optical density of water and the optical density of cornea is basically exactly the same. So the cornea has no ability to function in refraction underwater. And for the first 200, 300 million years of vision, there was no animal that even thought about vision above water. Vision was all below water. So everything was handled by the lens. And so if you have an animal that exists entirely underwater, it's a good bet that that animal is going to have an extremely round, a spherical, and also very dense lens. And that's the fish. I call that the fish solution. So this is a flounder, and that's typical of a fish lens. Not only is it spherical, but if you pulled it out of the eye, you could detect that it's is it over?
Oh, maybe now because
Just the
before.
exposure, yeah.
Oh,
Okay.
oh, now it's looking better now. Maybe it just took it a moment.
So this is typical of what you'd expect in a fish eye or any animal that has vision only underwater and doesn't care about vision in the air. That's not completely true. I'll talk about that. The curvature of the cornea then becomes irrelevant. Most fish have a flat cornea, and we'll talk about flat corneas when we talk about animals that want to see both above and below water. But the reason why a fish has a flat cornea is because it cuts down on hydrodynamics when it's swimming through the water. There's less resistance if it has a flat cornea.
It's the weeniest little iris.
Yeah. A little bit better on this side, but still pretty weeny. I don't know if there's anything special about the flounder. Notice that the corneal stroma has two components. One component that's continuous with the integument, and then there's another component that's continuous with the sclera. And so sometimes some people refer to the fish cornea as having a spectacle, so a two-part cornea. I've never really adapted that, but you'll see that in the literature.
Does it have epithelial lining of that space? or
No,
not?
no,
Okay.
there's no epithelial lining. Sometimes there will be a pigmented layer that extends into that space.
Mhm.
There's a bone. Many fish, not all fish, have a bone, and then they have cartilage. Like all vertebrates have a cartilage. It has an avascular retina, although there very often isn't membrana vasculosa, as I've shown in other species. There's no way that you're going to have accommodation by changing changing the curvature or the shape of the lens, because it's going to be spherical and dense. But they do, this is a skeletal muscle structure that extends basically from the ciliary body, attaches to the lens, and this is called the retractor lentis muscle, and it attaches to the lens and it drags the lens sideways and backwards. So in other words, retraction of the lens. And by moving the position of the lens, the fish is able to achieve some accommodation for near and far vision. Okay. The shark doesn't have a retractor lentis muscle. It also has a very round, dense lens, but it has what's called a protractor lentis muscle. And supposedly it moves the lens forward in position. I've never been able to find anything that I could recognize as a protractor lentis muscle in a shark. So I haven't pulled it to show you.
As they return from the fry stage to the flat stage and their eyes shift.
Yes. Their eyes are, they live flat on the bottom with both eyes on the same side of the head.
During that transition.
That's a great question. It's beyond my knowledge. That when the flounder is born, the eyes are positioned as they should be, and then as they develop and flat, the one eye migrates around the head, which is cool on itself. But there's no way I can show you that. So
I mean, it must be the entire skull that changes shape, right? Like the orbit must end up. It's not just that the eye comes floating around like in the sub-Q or something.
I agree.
Okay,
But I have
great.
no knowledge to share on that. Okay, the flounder is what we call a fin ray fish, a ray fin fish. And that's what most of the fish that we think of today, they have fins that have very thin bones and then a membrane that goes across it, and they use them to flap as they're swimming. A much smaller group of fish that are alive today are the lungfish, or they're also known as the lobe fin fish. And those are fish that have lobes rather than ray fin. They have lobes for fins and muscles in those lobes. Some of the lungfish that are alive today will actually walk on the bottom of the water. But in the Devonian, in the old days, when ray fin fish and lobe fin fish separated from one another, there was a whole variety of different fossils in the fossil record of lobe fin fish. And so, and many of them, rather than swim, they would use their lobe fins to walk around in the bottom. And then in the Carboniferous, some of those lobe fin fish, in order to escape predation or perhaps to take advantage of a new niche, would act, and since they have they're called lungfish because they have a swim bladder filled with air that they can also use as a secondary oxygen structure, analogous to a lung, and probably developed into a lung. So they're able to go up on land and crawl around to some extent on land, and that's how people think that land vertebrates, in other words, tetrapods, evolved from lobe fin fish. So today there are 3 species of lungfish, lobe fin fish, and they all are similar. They all have teeny tiny eyes. Coppola Lab has examples of all 3 of them, but they all look the same, and none of them are very well preserved. But I'm going to show you anyway. This is the section of a teeny tiny eye from a lobe fin fish. You can see already that the cartilage has become mineralized, and that's probably pathology. I'm not going to go beyond that to any extent. You can't really prove it in here, but trust me, they would have a round spherical lens because they have no interest in seeing above water. Well, actually, I don't know. Later on, some of them crawled up on lands and probably became something like an amphibian. But all three of the lungfish that we have similar retinas. Not a very, they have teeny tiny eyes, so they probably have pretty primitive vision. You can see that they have a shading of the photoreceptors, but they almost indistinguishable outer nuclear layer from an inner nuclear layer. They have pretty cool looking outer segments of photoreceptors. Including huge and prominent oil droplets. But that's all. And I think I showed you a an axolotl that has very similar morphology.
What's this? You tell. Okay.
It's an interesting structure in the. Okay, so we've been looking at vertebrate eyes. We looked at sharks. We looked at fish, lobefin and rayfin fish. We looked at a frog eye and a salamander eye, and then we looked at several kinds of reptilian and bird eyes. Does anybody know the most primitive of the extant of the still alive vertebrate specimens? They're actually not vertebrates, they're chordates.
Hagfish.
The hagfish would be one. Sometimes they're known as the jawless fish. The lamprey and the hagfish.
Do you have a four-eyed fish in your collection here?
We do have a four-eyed fish. Our gross picture of the four-eyed fish tells more than the
Okay. Okay.
histology. If anybody has access to a really fresh four-eyed fish, we'd like to have it.
We could buy one and kill it.
Then live peacefully.
The problem is that they rot so fast when they die.
That's
Very.
I think they have them at the Shed Aquarium.
Excellent.
I think you can get them
We'll
at.
ask one from them.
I think you can get them at the pet store. I don't think they're that common as pets. They what?
four-eyed fish.
Yeah, they have like a figure 8 shaped
Yeah.
like iris, right? Kind of.
Yeah, they have that.
Yeah.
They have optics for an upper to see above water and optics to see below water.
Sorry, carry on. Here we are.
So this is a lamprey, and this is so the lamprey has a biphasic lifestyle. For the first 4 or 5 years of its life, it exists as a larval form, and the larval form buries itself in the muck and feeds by taking in and taking out muck. So there's no light there. It has eyes, but they're buried in the head. And
Is this the larval form?
this is the larval form.
Ah, okay.
And they, for all intents and purposes, they look like an embryonal vertebrate eye. Now let's have a look at that. A couple of things to notice is that they are inside the head. The integument goes over the surface. Most of the retina, most of the neural retina is undifferentiated.
Cool.
The lens also looks like an embryonic lens with the lens fibers that go all the way from the front to the back, remains such. But there is a area in the central retina in the back, for reasons that I don't understand, does become differentiated. You can recognize photoreceptors. Notice that there's pigmentation only over a segment of the choroid, and I believe this is retinal pigment epithelial pigment rather than choroidal pigment. So that's the eye of the embryo of the larval lamprey.
And they're, sorry, completely covered with skin at all depths of the
They're
thing?
anatomically cryptophthalmic.
Okay.
And then this then is the grown-up lamprey. Did you get that hair thing out of there? So remember, this is the most primitive of the vertebrate eyes, and it's not really a vertebrate, it's a chordate, which means that it maintains a notochord rather than a spinal cord. Looks a lot like an underwater eye that we've been looking at. It has a spherical, probably very dense lens. Like a fish, most of the stroma of the cornea is derived from the integument rather than from the sclera. There's only a very rudimentary sclera at all. There's no cartilage, there's no bone. And again, it's looks like a fully developed vertebrate eye, primitive in many aspects, but pretty well developed. Interesting thing about this is this eye already, as the most primitive chordate eye, already has 4 different chromic variants of, so it's tetrachromatic like a bird, has 4 different color specific photoreceptor types. Okay.
Do they also have the retractor lentis muscle?
They do not have a retractor lentis muscle. I believe they have a retractor globi, retractor bulbi muscle, but I'm not sure. And I believe it's Heidi is our expert on the nerves of the central nervous system. I think it's innervated by the abducens muscle, abducens nerve again, but the retractor bulbi muscle of a hagfish of a lamprey.
Yes, retractable eyes usually abducens.
So this then is the other jawless fish that we have. This is a hagfish, and I'm going to try and find the eye. Oh no, that's the larval. Excuse me. Here's the hagfish.
Wow.
There's a notochord. I have to do some searching. Okay, so here's the eye.
That's it?
So here's the hagfish. Does anybody know anything about the hagfish? What the hagfish has a superpower. Does anybody know what its superpower of a hagfish is?
Producing mucus.
Mucus. It's a
Yeah.
it's a mucin specialist. If a predator grabs it, it secretes a copious amount of mucin. And I would recommend that you do some Google searching for
YouTube.
YouTube searching for mucus in the hagfish, and don't neglect YouTube searching for the great hagfish truck accident. It's an impressive amount of mucus. So the hagfish has no external eyes. The eye is embedded in underneath the integument, which is here. The hagfish eye has a retina, arranged like a vertebrate retina. But there's only 2 neural layers. Most vertebrate eyes have an outer nuclear layer, an inner nuclear layer, and a ganglion cell layer. This one has only 2 layers, and I don't know enough about hagfish vision to know what the names of the layers. Then there's a retinal pigment epithelium. There's an optic nerve, but I haven't. This section doesn't sample the optic nerve, and there's no lens.
There's no pigment.
There's no pigment.
Huh.
It's not a vision, it's not an eye that can accomplish vision because it's buried in the underneath the skin. And I guess you could call this a cornea. It's not, doesn't look like a cornea that I'm familiar with. We'll call this the vitreous. I'm not sure if that's accurate, but there are strands of stroma that go across that space. Now, the hagfish neuroretina, hagfish neurologists tell us, is
Neurologist.
connected to the brain in the same way that the pineal gland is connected to the brain in more advanced vertebrates. And the hagfish does not have a pineal gland. The lamprey does have a pineal gland, and obviously is a visual structure. So when I heard that, I got excited, and so I said, "Oh, that means that vertebrate vision is a derivation from pineal function. In other words, the detection of daylight length, and not designed for visual acuity or form recognition of form." So I thought, oh, the hagfish is basal, and the lamprey developed later. So, and I don't know if you know who Dr. Schwab is. Ivan Schwab is a a physician ophthalmologist who specializes in ocular surface diseases, but we know him because he's passionate about comparative anatomy, and particularly in relation to evolution of the eye. I passed around a book that he wrote called Evolution's Witness. So he was of the same opinion that the hagfish must be basal in terms of vision. But the geneticists tell us, and apparently it becomes more and more clear as more and more genetic studies are made, that the hagfish and the lamprey are sister taxa, and one is not more primitive than the other.
Oh.
So that means that this must be a degenerate eye that's lost structure, like I suggested for the mammalian eye or the mole eye, and has adapted to become nothing more than a daylight length detecting organ like the pineal gland would be in most mammals. I'm still open to the possibility that the geneticists are wrong and that this is a basal structure. I would love to be able to say that the vertebrate eye derived from a structure which is designed to just detect daylight length, but I'm apparently unable to do that. Almost every cell in the hagfish body is a mucus producing cell.
You mean in the skin?
Yeah, they have this, particular glands in the skin which are mucus producing super producers. Are people interested in a bathroom break? Okay, let's take a 10-minute bathroom break.
It has to do with the way they feed.
Monday at 9 a.m.
They like latch onto a fish and then they tie their body in a knot.
3308.
And they work the knot up to
Yeah,
the fish's body and then they use the force.
In my email,
pull a
I
chunk
would
of
my,
flesh off.
my,
Yeah, same with, I don't know about lamb, no, lampreys
There's
suck a
our
lot.
number there.
Yeah, they're parasitic.
But so I bet the mucus has to do with that.
Probably
Yeah.
could. Yeah. I don't think. I don't
One of the main
think
things that they feed on is whales.
Dead whales?
Apparently, when whales die, they
It should be
sink,
a
which is
problem.
probably a good thing.
You want to.
Yes. Right.
So experience already. There's nothing.
All right, so I'm running back.
Spend it cautiously and feel like you should get more. Experience them.
Yeah, sure. We have a journal club on Thursday. We do have lab meetings Thursday afternoon. It'll be fine. We're kind of gonna, but I think that's pretty much the only other thing. Other than that, yeah, probably be fine. Yeah, yeah.
And maybe I can also, I don't know.
Yeah, yeah, you figure it out. We're pretty lax, so. Sounds good.
you worked in a landfill lab one time, and we would stick our glove hands, we would stick your thumb in their little sucker mouth. And they would generally.
A couple folks had issues with parking, so Janice couldn't find parking, so she went home and got an Uber. And Andrew, I gave
So
him
the-
a Zoom link.
The lot is
The
full.
lot was full, yeah, so we might have.
When I came,
lost-
there were 17 spots. So, yeah,
Anyway, it was, they
maybe,
was just both. of
maybe
them
there
hit
were
the window,
people
right?
unwilling to do that.
Yeah, so anyway,
Yeah, because people came in, started coming in late.
yeah. Yeah.
There's salt water.
Janice was, so she went all the way back home and then called an Uber, so. That's dedication.
Would be like all in the place and everything. Yeah. They're like these troughs, like under grates and stuff, before like actually getting into that. Yeah. That local fish store, I knew shipping the fish.
Any, wide fish.
They don't, sadly. No four-eyed fish.
I wonder how many rare fish species you can get at stores here. But I grew up in Hong Kong where it's like the center of a lot of that training, unfortunately. So I used to have things like elephant nose fish.
We have we have elephant nose fish. Yeah. They have interesting eyes.
How's it different from?
Well, they're tiny for one thing. They're not, they don't have visual acuity. But they, my understanding is they want to see in cloudy water. So I'm gonna show you a fruit bat eye that has some similar features, but they have the retina is segmented so that each segment can function as an independent section to absorb a small amount of light, and then they function like an insect eye. And then they have a guanine tapetum.
I always wonder what's inside of that trunk-like structure.
And that I have no idea. Yes. Yes.
Branchies, which is like some sort of chubby goldfish.
Oh, yeah.
They're the sort of cheeks, yeah. Big
Hmm.
goldfish.
We don't have any of the really bizarre deep sea fish with tubular eyes or
Telescope fish,
the what?
telescope fish.
I don't know what that is, but we don't have one.
Yeah. Oh, barrel.
And we don't, a lot of people know what the mantis shrimp is. We don't have a mantis shrimp eye either. I cornered a scientist and made him promise to send us a mantis shrimp, but promise didn't take. The grocery store.
It's like
How-
in the live section.
In the live section.
It's like so next to the crab and lobster.
How do they keep it in fish tanks with how like punchy
I
they
don't
are?
know, it's fine, like, oh, yeah.
Cool.
I'd be afraid of like punch a hole in the fish tank. You can just make a mess.
I feel like if they were smart, they would try to punch the glass. I've never seen them try to do that. So,
Maybe they like do something like use rubber bands or something for.
no rubber band.
Oh,
They're just
yeah.
straight,
Okay.
straight,
Yeah.
like, sure. These scallop eyes.
So I'm gonna offer you an option here when I think we have at least one more person to come.
What do you call it? What's that snail that has the poison barb? Ah,
Cool snail.
Yeah, the one that stabs things. They have these eyes.
They die.
So
Yeah.
you have an option. I'm gonna go in, I'm gonna show you some specialized eyes for different niches.
We still have 4 more minutes.
Oh,
Please,
still,
sorry.
okay, thank you. I'll wait then. Till people get back.
Here, you didn't find.
No, I do not have a comb jelly eye.
They have eyes?
I do.
Really? Huh.
That's so fine. What is that?
There's one more person that's not here yet. Can you get them in the grocery store in Hong Kong? Not even in the live section?
Jelly, the ones that are edible are usually sand jellies, and they're dehydrated and like used in like cold fish.
Huh.
Can you buy them from Carolina Scientific?
Oh, I don't know.
Those comb jellies, or what are the
Mm.
ones you get in for the undergrad labs? You can buy them, roll
Oh,
little jellyfish, yeah.
Oh, come on.
Still think there's one person that's. Anybody that's not back yet, speak up.
we have 1 minute to go.
Oh, well, yeah, there's so many signal there.
I think
So we still have a variety of vertebrate eyes from different specialized niches, sort of like we've done on the last session. If I go through all of them and do them any justice, that's going to be the end. I also have a few invertebrate eyes, both molluscan and arthropod eyes that I could show. I actually know very little about how invertebrate eyes work, but they have interesting morphology if people are interested in looking at those.
Yes,
I hear nothing.
yes.
You want me to truncate the rest of the vertebrate eyes and,
Oh, no, keep going with vertebrates.
okay. Okay, so the next group,
Sorry, does anyone else, are they really interested in inverts out there?
I may, if nothing else, I may do the octopus and scallop eye.
Yeah.
Oh, yeah.
Yes, yes.
So this thing, we'll do these three in one. Vision in birds that dive underwater and actually swim underwater and catch fish underwater using their eyes. So we got your loons, you got your cormorants, you got your penguins, you got your puffins, you got your grebes, and then gannets is another one. We have in the Coppola collection, we've got eyes from most, if not all of those. I'm not sure. You also have diving ducks, and they're a little bit different. They're a little bit more like regular birds, but all the other ones I said have similar morphology. And I'm going to only show you one because the changes are the same. They're not all that closely related phylogenetically, So the changes that you're going to see are likely to be what's the kind of evolution? Convergent evolution. And we're going to do what we're going to do is a penguin. I should rub that penguin. Penguins, of course, are the most adapted for vision underwater. All of those birds have lifestyles that would cause them to want to have vision both above and below water. This is the penguin eye. So first of all, it has all the features you'd expect in a bird. It has a skeletal muscle, it has a bone, it has cartilage. There's a pecten, even though it's not sampled here. So in order to see both above and below water, there has to be some tricks played with refraction. It's going to want to see have refraction that would suffice for vision above water, and as well as suffice for vision below water. We've already talked about the fish solution. The fish would have, for seeing below water, would have a very round and very dense lens. Well, that's not the way any of these birds work. Oops, that's a low magnification. They all have lenses that are very far forward positioned and not particularly hard and not particularly round. So one of the things, and this is this is one of the reasons why I wish we could make an accurate measurement of corneal curvature. If I showed you the gross pictures of these eyes, it would be more convincing, but they all have relatively flat corneas. And so what would be the advantage if you want to see both above and below water to having a flat cornea, or at least a relatively flat cornea? What would that mean for refraction above and below water? If the cornea is not curved, then it's not going to participate in forming an image on the retina. So that means if you have a flat cornea, that means the optics are going to be the same above and below water. Now you can't have a flat cornea because the rigidity of the eye is dependent upon the intraocular pressure. You can approximate a flat cornea by achieving a very rigid sclera, and all of these birds have a sclera with a lot of interdigitating weaving and collagen, which presumably makes that sclera excessively rigid. So, but they all have a relatively flat cornea. So that means that once again, then, if you're going to have a flat cornea and the cornea is not going to participate in forming an image on the retina, that once again puts the onus entirely on the lens. But they have a malleable and relatively not a completely round lens. So I'm going to show you some features that are common to all of those birds. They have a Brooks muscle, the posterior ciliary muscle, which is robust. You can see it here, it's very robust. And then they have no, either that or very rudimentary, Crampton's muscle. If you remember, Crampton's muscle is responsible for corneal curvature. And if you have a flat cornea, particularly with a rigid sclera, there's no way that the Crampton's muscle is going to get the, by having attachments to the most peripheral and inner portion of the cornea, there's no way it's going to be able to change the curvature of the axial cornea. So it's abandoned the Crampton's muscle altogether, but it has a very robust Brooks muscle responsible for lens accommodation. And then all of these birds have a fairly rudimentary annular pad. And then the plica of the pars pliccata, like all birds, it attaches to the equator of the lens. It tends to attach a little bit rostral, a little bit anterior to the absolute equator. I don't know. I'm not exactly 100% sure why. So that's another, there's a number of features here: flat cornea, absent Cramptons, robust Brooks muscle, rudimentary annular pad, plica, which attach toward the front, and then they all have, remember I showed you in the turtle, they all have massive muscles in their iris. And it's been studied most in the cormorant. And in the cormorant, it's proposed that those features function together. It's not proposed how that works, but they squeeze the lens in such a way that the anterior curvature of the lens becomes dramatic, and it actually has the appearance of becoming entrapped in the pupil, so it bulges forward like that with a dramatic curvature. So it's interesting to me that they all have the same adaptations for vision, both above and below water, and that's what this robust muscle here makes me wonder if the turtle is not somehow similar. Okay, does that make sense? Somehow the diving ducks have eyes that are not that much different than other birds and don't have those adaptations. Okay.
Do you think that's because diving ducks also have to fly, whereas a penguin does not?
But
So they-
a puffin,
okay.
cormorant, and a gannet, and all the other ones.
Okay, sorry.
They use other senses when they're, I don't know, I guess that's.
I think it's because phylogenetically they're not that they haven't differentiated that long from other birds, but I don't really know. Okay, whales and sea lions. They're mammals that whose lifestyle would make it convenient for them to be able to see both above and below water. This is a beluga whale.
So tiny.
Yeah, I showed you the blue whale. That's so huge. I thought for a long time that this was a juvenile beluga whale, but I looked it up and it's not. It's a fully grown beluga whale. I'm not going to address that any further than that. They have fairly tiny eyes. They have the fish solution. They have a very round and very dense lens. They have almost no ciliary muscle for accommodation, and no matter how much muscle they had, they wouldn't be able to change the shape of the lens. There are people that propose that they have the ability to move the lens forward and backward, taking advantage of the structure of the eye and the structure of orbital musculature. And I don't have anything to say about that. Maybe they can move the lens forward and backward for accommodation. I don't really know. They also have a relatively flat cornea. Again, based on, you can kind of get the impression of that here, but based on either histology or gross appearance, it would be hard to prove that. But take my word for it, all of the cetaceans, all of the whales have a relatively flat cornea. They have cetaceans, if you'll remember, are artiodactyls. So they have
Slide up.
oh yeah. That's not the way a microscope works. I don't know if people have paid attention to how I was showing those very low magnification images. Thank you. But what you do is you use objective with a substage condenser in, and then put the slide on the illumination area. And That's a trick I learned from another veterinary pathologist. And so it's very handy when dealing with eyeballs, because looking at the very low magnification is good. One time I was at a meeting where there was a microscope salesperson selling his microscope, and I wanted to show somebody something in a slide. So I did that. I put the slide down below and adjusted it, and the salesman came running over. He said, "No, no, no, that's not how you use a microscope." So I had to show him what I was doing.
You mean I've been doing it wrong my whole career? Wow.
So relatively flat cornea. The other thing that's common to all of the cetaceans is that they have this extremely thick posterior sclera. And again, if you look at the way the collagen is arranged, it's like the limbus in that penguin. They're interwoven. So not only is the posterior sclera thick, but it's also very rigid. And this is where people say, oh, well, that's because they have to dive deep in the water and they have pressure. I don't really have much to say about that. I just don't think that's likely to be true. And it's very much argued what the function of this thick sclera is in cetaceans. Chris Murphy, some people here know Chris Murphy and some people don't. He and I were largely inseparable when he was here. And he is an independent thinker about comparative anatomy and how things work. And he came up with the idea that the cetacean eye is usually behind is often behind what's called a melon, which is an acoustic device to focus acoustic energy for echolocation. And so that means that the whales use echolocation, and they produce an extremely high decibel, extremely low frequency sound in order to communicate with one another over long distances, or to use echolocation. And he proposed that this thick and rigid sclera was acoustic protection. And I like that idea because I do believe that high decibel, low frequency sound is at least potentially toxic to the retina. But all of that is just speculation. It's not science by any means. So here's I mentioned earlier that the whales and pinnipeds both have a tapetum. The whale is related to the hoofstock and would have a fibrous tapetum, whereas the pinnipeds are carnivora and would have a cellular tapetum. But the thing of it is the tapetum covers the whole back of the eye because they go deep in the water and they need to have the dim light vision both in the upper and lower portions of the visual field. I have to kind of cherry pick the retinas here. A feature of the artiodactyl retinal vasculature is that the large retinal blood vessels will kind of bulge out into the vitreous. And just another feature that suggests that yes, indeed, the cetacean is derived from artiodactyls. So if you're a photographer, and you have an eye that's probably not capable of accommodation for near and far vision, and you want to catch fish in your mouth, so you probably want to see things that are fairly close. What's another way other than changing the structure of the focusing features of the eye? What else can you change, particularly if you're a photographer? You want to take a picture of something and you want what's near and what's far to be in focus at the same time. What can you do?
Aperture.
Yeah, you can ramp down the aperture so you have something approaching a pinhole. You can get an image that's nicely in focus if you have just a pinhole at any distance, so both near and far. So if you can achieve a pupil that's sort of like a pinhole, then that will, in bright light anyway, if there's enough light, then you'll be able to see things that are both near and far. I've been in the beluga whale exhibit, and I know that they stick their head above water, and the trainer will throw them a fish, and they'll catch a fish. So I know they can have some vision above water. And unfortunately, I'm not going to be able to show you the cetacean pupil is like a horseshoe. It curves around, and that horseshoe component is called the operculum. In dim light, or when they dive deep in the water, the operculum goes away, and they have a nice widely dilated pupil. But in bright light, the operculum closes, and they have, for all intents and purposes, a filled-in pupil with two pinholes where the operculum meets the rest of the cornea. So you can see that histologically. This, I believe, is the lower portion of the iris. I can't tell because they have a tapetum on both sides. And this, I think, is the upper where the operculum is. Both sides have a very robust sphincter muscle, but this one is more prominent. So they, in bright light, they can achieve accommodation by achieving something like a pinhole pupil. That's the cetacean. The pinnipeds, this is a California sea lion. Also rely on the fish solution for seeing above and below water. They also have a flat cornea, a relatively flat cornea. The pinnipeds, at least the California sea lion. Oh, there's one other thing I want to show you in that pinniped, in that cetacean. The pinniped, let's go back and look at it. There's one other cool feature in the whale eye. If we go back in the eye to the optic nerve, and if we look on either side of the optic nerve, there's a rich collection of arteries on both sides. It's just arteries, it's not veins. Any idea what function that is? It functions as a little heater. It concentrates heat in this portion here, which then causes the eye to be at a little bit higher temperature than tissue around them, which gives them a little bit more visual advantage when they go deep in the super cold water. Deep hunting predatory fish don't have a what's a collection, a tangle of blood vessels called. That's not really a reedy. A what?
Plexus.
Plexus. Don't really have a plexus of blood vessels like this, but they have extraocular muscles that shiver to produce heat when they go deep. The pinniped also has a relatively flat cornea. Some pinnipeds, this is a California sea lion, and the California sea lion is one of them. They actually have a plateau in their corneal curvature that may actually be totally flat. I'm not sure, and it could be that plateau is used for both above and below water vision. I'm not sure about that. They have, you wouldn't believe it from looking at this specimen, but yes, indeed, Keep doing that. It's not the way you use a microscope. Pinnipeds have a relatively flat cornea. They also have a very rigid sclera, which is probably why how they're able to achieve that relatively flat cornea. They have they don't have an operculum in their to control their pupil, but rather than a round pupil, they have kind of a pear-shaped pupil. And if you have a round pupil, you're limited into the extent to which you can contract that pupil. If you have an irregularly shaped pupil, then your muscles can be arranged around that irregular pupil in order to close that pupil more. So the pinnipeds are able to achieve more or less a pinhole pupil. But again, that's only going to be useful to them under bright light. And if you go to the zoo where they have pinnipeds and you can buy some fish, again, you can throw them a fish and they'll quite easily catch the fish above water. They have a very round, very dense lens. Like I said, it's the fish solution. So they probably can't change the shape of their lens for accommodation. And they probably don't have much ability to move their lens back and forth, but they do have, I'm not going to bother to try and show it, but they do have more robust and more complicated smooth muscle structures, both in their iris and in their ciliary body. Maybe they can use those muscles to move their lens back and forth. I've never seen a study that has tried to examine that. So these are carnivores related to dogs, bears, etc. And you'd expect then would have again a tapetum that goes both in the front and the top and the bottom portion of the fundus. They have a cellular tapetum. It looks for all the world like a dog tapetum. And they too have a plexus of arteries adjacent to the optic nerve, but not as robust as the ones you'll find in the cetaceans. Any questions about marine mammal vision?
People shapes, so I was thinking about goats, like horizontal.
Animals that have a horizontal pupil are largely animals that live in the in the big sky area with open horizon, and the horizontal pupil helps them to see widely in the horizontal plane. Okay, this is the eye from a bat. It's called the little brown bat, which is one of the common bats that we have in this area. So it's what we would call a microbat. And microbats, as you probably know, catch insects on the fly, not using vision, and at night, not using vision, but by using echolocation. But they do have eyes. They have teeny tiny eyes. But they're fully developed eyes. They have everything you'd expect to find in an eye. They have a differentiated lens, a differentiated cornea, a retina that has all of the features of a nocturnal retina, a very thick outer nuclear layer, and very few ganglion cells. They do not have a tapetum. And another feature is, for some reason, they have a very pigmented sclera. I have no idea why that is. Another feature that's not illustrated here is they have very robust meibomian glands, but that's not sampled here. So why do they have such tiny eye? Well, my question is, why do they bother to have eyes altogether? Because echolocation provides them a lot of sensory, provides a lot of their sensory needs. But I think they navigate by using the moon, and you can't echolocate the moon. And perhaps also very large structures in their visual field. They need to have vision, and so they need to preserve eyes. They probably need to have such a teeny tiny eye because their head is full of acoustic equipment and there's just no space there and to keep a balance when they're flying. So they do have teeny tiny eyes. I say blind as a bat, but I suspect that they don't have great vision, but they definitely do have all the structures you hope to have. So next I'm going to show you a fruit bat. Fruit bats have relatively large eyes. They don't have echolocation. For the most part, they're nocturnal. And they have a very interesting feature of the eye, which I'm going to show you, and you're going to identify when you see it.
Did
This-
they ever have echolocation?
I don't think so. They're derived from one another. I don't know if the microbats with echolocation are more differentiated, more derived than the fruit bat or not. That's probably known, but I don't know. Okay, so here's the fruit bat. I'm going to sneeze. They have eyes that are much like you'd expect for a nocturnal eye. They have a big wraparound cornea. They have a very round lens, which is more posteriorly positioned in the eye. So adaptations that we've already talked about for nocturnal vision. And I'm going to show you. That's not the best preserved specimen in the world. The retina is kind of fractured, but what do you notice about the back of the eye? It's corrugated. And the part that's corrugated is the choroid. So, of course, there's a lot of confusion among scientists trying to figure out, well, what the heck is going on here? I came up with the opinion, and then I found an article that agreed with me, so I'm sticking with it. Remember how I said, and then in nocturnal vision, the goal is to recruit many photoreceptors, rod photoreceptors, so that some of those photoreceptors in the dim light, some of those photoreceptors will make some kind of a connection, a neural connection with the limited amount of light that's available, the small number of photons that are available to converge on a small number of ganglion cells. So that's the way I think this fruit bat retina works, that each one of these little valleys is a functional unit, so that any light that comes into it and gets into this area will converge on a small number of ganglion cells and produce some kind of an image. In other words, it's a lot like a compound eye in an insect, so that the vision will be a kaleidoscope of different functional units. Not very great for visual acuity, but potentially good for visual sensitivity. Now, the retina, generally speaking, doesn't have retinal blood vessels. However, each one of these little hillocks has a blood vessel that comes in and then goes back in again. So another theory that some people have is that this arrangement is to provide nutrition for the inner retina. And I'm just saying that's not my favorite theory about this. I'm not going to be able to show you in this specimen, but the fruit bat has a tapetum. It has a retinal tapetum, and I'm gonna go down on it, even though it isn't obvious in this specimen. If we look at the retinal, the pigment in these hillocks is all choroidal. It's not in the retinal pigment epithelium. Here's the retinal pigment epithelium. And if we had a good specimen, you'd see that the retinal pigment epithelium cytoplasm is filled with foamy oil droplets, and those foamy oil droplets provide what we call a retinal tapetum, or in other words, a white tapetum, to reflect light back through the retina. So I'm going to show you an opossum eye, which also has a retinal tapetum, and it's going to be more easily recognized than it is in that fruit bat. Opossums are New World marsupials. What?
Wondering, so if the fruit bat's anangiotic, but has the corrugated,
Correct.
yeah, corrugated choroid. I mean, if it was to do with vasculature and blood supply, I mean, do other anangiotic species have the corrugated?
Yeah, so we've already talked about the membrana vasculosa in fish and amphibians, etc. We've talked about the conus papillaris in reptiles and the pecten in birds. Many, I hesitate to say all, but many marsupials have what's the word you gave me, Kelsey? Plexus of blood vessels that, not like a conus papillaris, but bulges out from the optic nerve into the vitreous that perhaps helps to provide nutrients to the inner retina. So those are some examples that come off the top of my head. And I don't think we have an optic nerve in this opossum. I think we have a better section. Yeah, here's a better section. Unfortunately, we don't have an optic nerve. So again, it's a classical nocturnal eye with a big wraparound cornea, a more centrally positioned round lens. We're interested in, only thing we're really interested in here is the tapetum. And if we go on this side, you can see that the retinal pigment epithelium has pigment, and I think if we go even further, it becomes more cuboidal. And if we keep going in this direction, the pigment will disappear, and the cytoplasm of the cells will be very thick and kind of foamy appearance. And histologically, ultrastructurally, that's a whole bunch of little oil droplets. And if you do fundoscopy on these, you'll see that the back of the fundus is white like a piece of paper. And if you've driven around at night and you've got your headlights on a opossum, you can see that it has a bright tapetal reflection. I didn't realize this for a long time, but South America is loaded with marsupials. There's a whole bunch of different kinds of marsupials in South America. And in fact, Australian marsupials migrated to Australia across Antarctica from South America. So the differentiation of marsupials happened in South America. We have a planted ophthalmologist interested in comparative anatomy and lives in Brazil. And he promised to send us as many marsupials as he could from South America. All the ones that he sent me are basically opossum clones. They're exactly like opossums. They have a retinal tapetum as well. So I don't know if that's generally true of the South American opossums. The Australian opossums rarely have a tapetum. The koala has a fibrous tapetum, and I don't know if there are any other Australian marsupials that have a tapetum. What time is it? Oh, 4:30, man. So we have time to do the squirrels. So this is a ground squirrel, a 13-line ground squirrel. The ground squirrel is a rodent, and it's diurnal. It lives in its burrow at night and comes out during the day. And so you can see it has a lens. It's got a small eye. So having a small eye, it pretty much has to have a big, more or less round lens. So it becomes a little bit hard to identify a nocturnal from a diurnal animal based purely on the shape of the lens. But I think you can buy that the lens is pretty far forward in the eye compared to the size of the eye. The cornea is not a wraparound cornea. It's relatively small. And then here's the retina in back. And they have a prominent retina. And we're going to pay more attention to the retina. I think we've already talked about that for a long time it was thought that the 13-lined ground squirrel had a pure cone retina. They have a very diurnally adapted retina. Well, that doesn't turn out to be true. They have both rods and cones. So here's the optic nerve. And we're going to center in on that area.
Is that artifact going? It's kind of smooshed up against the back of the eye. Sclera.
The nerve. You mean the direction that the nerve goes? I'm not sure.
The ground squirrel has a really long, flat optic nerve head.
Oh, kind of like a rabbit.
I have no idea.
No.
But I know the mouse is
Yeah,
like,
different than a
as
rabbit.
a whole, but it's like a really long, it's
Okay.
kind of, it's very bizarre when you look at it.
Okay.
Yeah.
So you're 3 steps ahead of me.
That rings a bell.
So using the principles that we talked about, diurnal versus nocturnal retina, there are lots and lots of ganglion cells, and the outer nuclear layer is relatively thin, making you think that it has a large amount of cones and not so many rods. I told you, as if I was telling you the truth, that mammals do not have shading of the photoreceptors. And it's granted, it's not as dramatic in the ground squirrel as it was in the birds or the fish. But I think you can kind of get the impression that the teeny outer segments of these cones might possibly be embedded in this very robust pigmented retinal pigment epithelium. And so I like to say that the ground squirrels are the exception, that they derive considerable advantage from being able to shade their outer segments of their photoreceptors. And the other thing to notice, as was mentioned, here's the junction between the retina and the optic nerve. We can call it the optic nerve head, and it's extremely small. It's extremely narrow. This eye is sectioned in the vertical plane as we usually section mammalian eyes. We try and section them in the vertebrate plane, largely because we want to sample both the tapetal and the non-tapetal retina. Well, the ground squirrel doesn't have a tapetum. Instead, it has a retina like this. Now, we're going to look at something else. Here's where the optic nerve is. And we're just going to pay attention to the distance of the retina on one side. We'll start with this side. The retina goes to about here. And now we're going to look on the other side. And the retina on this side goes to about here. So it's asymmetrical. It's much longer on this side than it is on the other side. So now you're going to tell me which is the upside and which is the downside. Where does the 13-line ground squirrel want its concentration of vision to be? Up or down? Up, yeah. So the sentinel ground squirrel is charged with protecting the colony when they're grazing in the prairie. And so that squirrel's job is to keep an eye out for predation, and the main source of predation is going to be from raptors above. So it wants to see up, and that means that the bulk of the retina is going to be down for looking up. Okay, now I'm going to show you another rodent with a similar lifestyle. But this eye is cut in the horizontal plane. And there's the optic nerve head cut in the horizontal plane. And if you look carefully, you can appreciate that in this plane, it's a series of little fenestrated structures, each one of which is more or less the same as the as the junction of that 13-line ground squirrel. So this is a chipmunk, also a ground squirrel with a similar life habits. And as has been said, the optic nerve head in the ground squirrels is elongated horizontally with a streak. Tree squirrels, which are also diurnal, but they live in trees and probably don't have to be so wary of raptor predation, have an elongated optic nerve head, but it's more oval than linear. And then a second. Oh yeah, let me take a look at this. I thought I threw in here the flying squirrel is also a squirrel. It's phylogenetically related to the squirrels, but it's exquisitely nocturnal. You're never going to see a flying squirrel around here because they're only out at night. And it has a round optic nerve head and a very exquisitely rod-rich retina. I think there's two things I tell the squirrel scientists they should study. One is the reaction of the ground squirrel retina to extremely bright light, and the other is that the genetic differences of vision in the flying squirrel versus the ground squirrel. Okay, you ready for some molluscan eyes? I have a few specimens that illustrate the importance of lifestyle for the integrity of optical structures. I have a comparison of the rabbit versus the grizzly bear eye, and then I have two different species of Congo river fish, one from that lives deep in the water that has degenerate eyes, and one that lives superficially that has normal fish eyes. And if I wasn't eager to show invertebrate eyes, I would show those, but maybe another time. What do you know about the octopus eye? The lens is split in two. That's very good.
The retina is backwards.
The retina is backwards. So the photoreceptive cells point forward in the eye. They what? They don't have blind spot because there's no optic nerve head. The optic nerves traverse from the eye to the brain in the back of the eye. They have the optic nerve, rather than going to a central brain, the optic nerves from each eye go to an optic lobe of the brain. So each eye has its own optic lobe, and that's connected to a central brain, but each eye has its own optic lobe for vision. And there's a cool thing about the cornea.
They don't have one.
Many of them don't have a cornea. If they have a cornea at all, it's very attenuated. And also, I'm not sure if this, I hate to use the word always, but they either always or often have an incomplete cornea. So that allows seawater to go behind the cornea and bathe the front of the lens. So this is an octopus eye. It's what's called the common octopus. Doesn't fit on the stage here. So there are three phyla that have eyes that are capable of something that we could call visual acuity. One is the mollusca, and I'm going to show you probably the most advanced eye for visual acuity. The other is the arthropod eye, and I'll show you what I think is probably the most advanced arthropod eye for visual acuity. Although, well, then I'll show you two eyes that are differentiated in different directions for visual acuity. And the other, of course, is the vertebrate eye. So here's this is a common octopus. This may be jittery. Well, not gonna try and do that. You have to move this around quite a bit, but here's the front of the eye. This is the integument. And so here's the cornea. And even at its best, it has a very rudimentary cornea. And I don't know specifically about the common octopus, but I think that most of, or perhaps all octopuses have an incomplete cornea, so that allows seawater to be both here and here surrounding the lens. It's a transparent cornea. This structure is known to octopus anatomists as the iris, and it's in a position much like the iris in a vertebrate eye. This structure in back of the iris is called the ciliary papilla. It's in many ways analogous to the ciliary body, and it's an epithelial structure. with an epithelium both on the front and the back. And notice that emanating from the epithelium, there are these cords of pink material, and those are in fact lens fibers, and they become incorporated in the lens. So the ciliary papilla is producing the lens, and it's producing the lens by making two different portions of the lens. Here's a portion and here's a portion. Octopus anatomists call this the external lens and this the internal lens. In my simple mind, I call this the anterior lens and this the posterior lens. But it's secreted by the ciliary papilla. There's no lens capsule. The lens proteins, the lens fibers, are made up of crystalline proteins of the same class of protein as the crystalline fibers in the vertebrate lens. But I don't know much about crystalline protein chemistry, but apparently it's a different class of crystalline protein. Why does it have a two-phase lens? I'm not sure. Scientists that make lenses for cameras or attempting to make accommodating lenses for intraocular prostheses have experimented with a biphasic lens like this in order to achieve some accommodation. I have no idea whether there's any advantage to that along those lines for the octopus or not. And then if we go towards the back of the eye, here's the retina. And the photoreceptive cells of the retina point forward in the eye. And then you can see in the back, it's a gamish of different kinds of neural populations. So I guess equivalent then to the inner and outer nuclear layer and the ganglion cells. And then notice that there's a pigmented layer in the middle of the retina, right where it should be, right in back of the photoreceptor part of the retina to cut down on glare. So people say, well, the vertebrate retina is designed wrong because you want to have the light hit the photoreceptive portion of the retina first, and so that the molluscan eye is designed appropriately, I say that that's not necessarily true because as we've covered several times, the photoreceptor cells are the most oxygen dependent cells in the body. And if you have the photoreceptor cells positioned in the back of the eye, then they can be next to a rich capillary bed of blood vessels. If they're in the front of the eye, there are blood vessels that go into here, but they're relatively less significant than they would be in the vertebrate eye. So I question whether or not there's any advantage. The photoreceptor cells of the mollusk, that is to say the octopus, the squid, and the cuttlefish, are monochromatic. There's only one visual color receptor, so they see things in black and white. And then the nerve fiber layer comes off the back of the retina and forms these into these little optic nerve, I call them nervelets. And the little optic nervelets will traverse. Here's the cartilage. Here's the sclera. The sclera is made up of cartilage. It doesn't look exactly like cartilage to me, but octopus anatomists call it cartilage. So who am I to complain? And then they traverse through the sclera and then recongregate on the other side of the sclera and continue going until they congregate in this brain structure here, which is the optic lobe of the brain, where vision is processed, or at least it's first processed. Now, the integument of the octopus, that is to say the tentacles of the octopus, and I don't know anything about neural processing in the octopus, except that supposedly the tentacles are able to process sensory information as if they were their own independent sensory apparatus. So how that works with an eye and integumentary sensory system, I can't even imagine. There's another structure you may have noticed within that choroid, and that's this structure. This structure, you'll find it in all three, the octopus, the squid, and the cuttlefish. And it's called the white body. If you see it in the gross eye, you might be convinced it's white in appearance. And this structure, if you go to high magnification, you might guess at its function. It's made up entirely of round cells, and this is a hematopoietic organ. There's only one cell in the molluscan blood supply, and that's a cell called a hemocyte, and which is basically I'm way out in deep water without a life preserver, but it functions as an immune cell, probably like a phagocyte does. There's no red blood cells because the oxygen carrying protein in the molluscan bloodstream is dissolved in the serum, and it's made up of not an iron containing molecule, but a copper containing molecule. So that makes the blood of an octopus blue. And so I've exhausted just about everything I know about. I'm not going to go into it, but the other superpower of the octopus is its camouflage ability. And if I tried to explain how the camouflage function works in the integument, I'd have to go back and read about it before I did. But there's a very complex system of different kinds of cells that are capable of changing color in the integument of an octopus. So here is another cephalopod. The octopus, squid, and cuttlefish are all cephalopods. So this is a squid. I'm going to see if we can get this there's nothing under high magnification that I particularly want to show. Yeah, that works. So this is a section through the head of this squid species. Includes both eyes and both optic lobes of the brain. But there's an interesting feature, and they have both the octopus and the squid have the fish solution with regard to focusing light on the retina. They have a very round, very dense lens, albeit a two-phase lens. And you can see the nice ciliary papilla here and here. But what do you notice about this head?
Bigger.
Yeah, it's asymmetrical. It has a big eye and a little eye. And this is a squid that's called the cockeyed squid. And Dr. Schwab says, and I believe him, that this is the only animal that has different functions for the right and left eye. So here's the top of the squid. And so the big eye, if you look, most of the retina is down below. And so this eye is, functions for looking up. And this eye is used for seeing the silhouette of prey species of fish in the water table above the squid for predation. And the little eye, there's not that difference of upper and lower. And this eye is used for looking at depth, and it's used for looking for bioluminescence at depth. And bioluminescence is a signal for prey. So it's to look out for prey species, not prey, predator species. So who has the highest degree of visual acuity relative to body size? Any guesses?
Dragonfly.
The dragonfly, in my when I do this, has the largest eye relative to body size.
Spider?
The jumping spider. So here's the eye of a jumping spider. And if you're familiar with a jumping spider, oh, so dirty. The jumping spider has 8 eyes.
I've just heard they're the new hot pet to have.
Interesting.
Is it jumping spider?
Yeah.
What do you do with it?
Watch it.
Love it. Give it things to eat. Watch it jump.
Did you? I just caught it from my window.
What'd you do with it?
Just watch, oh, feed it like mosquitoes and.
So the jumping spider is a predator, and it catches insects by jumping on them, catches things by jumping on them. So it has these gigantic eyes that point forward, and you can see them. They have the appearance of being like a telescope. They're very long.
Is this sort of upside down right now?
This is
I'm
cut
throwing
in
out
the
myself.
in the coronal plane of the head. So here would be the front where it's looking. Here's the back of
Okay.
its head.
Okay.
On one side of this would be the bottom, and the other side would be the top of the head.
Okay.
So the primary eyes, the big eyes, look forward, and they're like binoculars. They have stereopsis, so they have depth perception. And then the retina, for all intents and purposes, is just one big fovea. And it's a complicated multi-layered structure, and has different color sensitive photoreceptive cells. But that gives it visual acuity. And then it has 2 other eyes that face forward. These eyes that are off to the side of the primary eyes are the second biggest eyes, and they're like a finding scope on a telescope. since these eyes have very, very narrow field of view, the spider wants to orient itself so these eyes are both focused on something. It has to do that by turning the whole spider around. So it uses these like a finding scope and a telescope. And then the eyes in back are more primitive eyes, and they're just detect potential predators. And they have, if you look at a graph, visual acuity versus eye size, the jumping spider is off on its own with a high degree of visual acuity relative to body size.
So these are the lenses, right?
So there's the lenses. The lenses is part of the integument. It's part of the chitinous integument of the spider.
You have 3 minutes, Dr. D.
Okay, I only have one more to show, and that is the eye that has the biggest eye relative to body size. The dragonfly. If you haven't, you should watch a Google. And if you're scared of jumping spiders, you can solve that by Googling a YouTube video of the mating dance of the peacock jumping spider.
Dude.
So this is a dragonfly. And it's not the greatest specimen in the world, but here's the head. Here's the dorsal midline. So all of this wrapping around the head is all the right eye or the left eye. And the insect eye, the arthropod eye, as you probably know, is a compound eye. So rather than being one camera-like eye, it's a fusion of myriad individual light sensing organs called ommatidia. And so each one of these subunits is an ommatidia. It's covered on the surface by the exoskeleton of the dragonfly, the chitinous exoskeleton. And then there's always a bulging area in the front of the ommatidia, which is referred to as a lens. It's not so much that it focuses light, but it makes sure that any light that comes here is going to go into only one ommatidia. It won't cross over into other ommatidia to degrade the vision. And then the body of the ommatidia goes all the way from here to here. And within the body of the ommatidia, there are 7 photoreceptive cells, including 4 different color variants and 3 dim light cells that are equivalent to rods. So again, they have tetrachromatic vision. And then at the back of the ommatidia, once again, it passes through a fenestrated chitinous layer. And then you can think of this like a little optic nerve. Those fenestrated nerve structures traverse directly into you can see it up here, but you can't see it down there, directly into this structure, which is the optic lobe of the arthropod eye. And we go to lower magnification. You can see that in the planar section, we only have the optic lobe contiguous with the eye in this portion. It would be contiguous all along here. So it's a very complicated laminated structure about which I know nothing, except it's the first line of visual processing. And then there's a central brain as well. In this planar section, it's a very underwhelming central brain. I have some other sections of insects where the central brain is much more prominent. So I don't know if the dragonfly does in fact have a rudimentary central brain, or if that's just a feature of the planar section. These open areas here are called tracheals, and they are connected to pores in the cytoskeleton, which allow air to circulate like air ducts. So that's oxygen supply for the tissues, because there's no pumping blood or blood pumping heart or anything like that. At least I don't think there is. And the other superpower of the arthropods is the speed of their neural processing. The dragonfly is a predator, and if you study predators and you study the percentage of time when they attack, when they go after something, and the percentage of times in which they're successful, the dragonfly is the champion among all predators in the world. It wasn't me that studied that, it's somebody else that studied that. But supposedly the dragonfly is the champion, the most successful relative successful predation attempts compared to the total number of predation attempts. And one of the reasons for that is they have such rapid neural processing. And if you try and catch one, like any insect, the reason you can't catch them is because their neural processing is so fast that basically when they see something, they're seeing things in slow motion. So you can't move your hand fast enough to be able to catch them. And that's one of the reasons why they're so successful.
Slow to stop somewhere, and you could go really slow to reach towards it, it won't fly away. Is it because if your slow motion is like extremely slow?
Do they have,
Barely moved into.
are they in the live section of the grocery store? Are they in the live section of the grocery store?
Wow.
Tricks to be slow. Same for butterflies.
Yeah, same with flies.
Mhm.
or anything.
Yeah.
Okay, we're all tasked with a chance. Yeah. So we have a dragonfly with eye pathology. One of our residents we used to take our eyes for processing to the medical school, which meant that our fellow had to carry eyes from here all the way to the medical school. And on her trip one time, a dragonfly landed on her arm, and she was able to notice that the dragonfly had a black spot on its eye. We have pictures of it. So she knew just what to do. She captured
Killed
she captured that dragonfly until it was formalin available, and we have the eye pathology from that specimen, which is interesting. Okay, that's it.
Yeah.
And does anybody have a favorite? Among all the eyes, a favorite? I was forced to say the chameleon was my favorite. The octopus. A lot of people like the octopus.
It's like the platypus.
The platypus.
Platypus.
Yeah, particularly in its relationship to evolution of the mammalian eye.
Like
The what?
the nautilus.
I didn't show the nautilus.
Yeah.
So these guys that sit around the Coppola lab, and every time we have a visitor, I force them to sit through hours and hours and hours of comparative anatomy, and I go through this same spiel. So they're all familiar with the spiel, but they've never actually done it with the slides. So Liam
Listen.
has missed the nautilus section. Any others? Okay.
All right. Thanks,
Thank you.
everyone.